Welcome to Catch This. My name is Trudy Ray, and today I would like to talk about the new variant of SARS-CoV-2 that is currently circulating in the UK. You may have noticed that all throughout this pandemic, there have been headlines warning that the virus is mutating to become more lethal or to spread more easily. We know that viruses do indeed mutate, and that is normal. It happens all the time. But when are these mutations functionally meaningful? And when do we get a different strain as opposed to just a different isolate or variant? I will attempt to answer this question in this video. Coronaviruses do have a proofreading mechanism that corrects errors during replication. However, this mechanism is not perfect, so occasional mutations are acquired in the viral genome. As I mentioned in a previous video linked here, the SARS-CoV-2 genome consists of RNA, which is then translated into protein. For translation to occur, the genome is scanned and read in triplets of RNA nucleotides. Each triplet codes for one amino acid, and as you may remember, amino acids are the building blocks of proteins. However, there are several triplets that can code for the same amino acid. For example, the triplets GCU, GCC, GCA, and GCG all code for the amino acid alanine. If a mutation occurs that changes the last nucleotide, U, in the triplet GCU to a G, you end up with GCG, which still codes for alanine and does not result in a change in the protein. However, if the mutation changes the C at the second position of GCU to a U, you get GUU, which codes for a different amino acid, valine. This will result in a change in the protein and may sound alarming, but in fact probably still won't change the function of the protein. That's because alanine and valine have very similar physical properties. Now, if the mutation changes the G at the first position in the triplet to a C, you get CCU, which codes for the amino acid proline. Proline is known to change how a protein folds, which could affect the ultimate function of the protein. However, that may still not necessarily happen. It is actually quite difficult to predict exactly how an amino acid change will functionally affect a protein. And even if it does have an effect, this effect may only show in cell culture, but may not show in humans. Such is probably the case with the amino acid change that has been hyped in the news for several months as supposedly making SARS-CoV-2 more transmissible, where a D amino acid, which is aspartic acid, changed to a G amino acid, which is glycine. So far, this effect, the increased transmissibility, has only been shown in cell culture. And until an amino acid change has a clear effect on the properties of the virus, as in it confers a different function on how the virus replicates or binds to host cells, for example, we can't call it a different strain. It is still the same strain, just a different isolate with a slight mutation in its genome that has no bearing on its function. So what makes a new strain? A new strain is a virus with a new biological property. Such properties might include an increased or decreased ability to bind to the cell receptor, so the virus can enter the cell, or an increased or decreased ability to replicate, or an increased or decreased ability to assemble new viruses in the cell. So there is a new isolate circulating in the UK named VUI 2020-12/01, which stands for First Variant Under Investigation in December 2020. So what is going on with this new variant? Is it a new strain? This particular variant is defined by multiple changes in the spike protein. However, the exact effects of these changes have yet to be determined fully. Nobody has done any experiments to determine the effect of these changes on any property of the virus. So why is everybody saying that it's more transmissible and more dangerous? According to the international research journal BMJ, on December 14th, England's Health Secretary, Matt Hancock, told the House of Commons that initial analysis showed that the variant may be associated with the recent rise in cases in the UK. The problem with making such a statement publicly is that the press will immediately pick it up and probably translate this phrase into "is associated with." The thing is that there have been no new properties ascribed to the new isolate circulating in the UK, other than sequence differences, which is not enough to make a new strain. It's just a different variant. There are a few variants circulating because that is what viruses do. They mutate all the time, especially RNA viruses. Just because a variant is associated with increased rates of COVID-19, this is still just a correlation, and we can't really say that this new variant caused an increase in infections. More likely, what we are observing is probably a phenomenon called the founder effect. Founder effect is the reduced genetic diversity, which results when a population is descended from a small number of colonizing ancestors. Using this graphic to illustrate the concept, think of each of the red and blue dots as a single virus. A single red virus or blue virus that ends up in a new person will be the founder. What could have happened is that one person who was infected with this new variant, let's say it was the red one, went to some sort of big event and infected many other people. You might call this a super spreader event. If you have 2 or 3 super spreader events like that, that virus will end up being the predominantly circulating virus. It really is just a matter of luck for the virus. Then it becomes the predominant variant, not necessarily because it is so much better at infecting, but because that is what is currently circulating. But the media may look at this and interpret it as a new mutant that spreads more easily without any evidence whatsoever. And this type of information makes the rounds very quickly, particularly by headline, because it is alarming and sensationalist. To prove that this variant spreads more easily, you would have to do the right experiments, and those experiments have not been done. At this point, it's all speculation. Now finally, let's address the elephant in the room. Will this variant render the new vaccines ineffective? In my opinion, that is highly unlikely. The vaccines that are currently in use or are about to be used encode the entire SARS-CoV-2 spike protein, and there are many immunogenic regions called epitopes on this protein. After vaccination, our immune system should produce a range of antibodies and T cells against these epitopes, and it is unlikely that these amino acid changes would render current vaccines ineffective. That's it for today. Thanks again for listening. My name is Trudy Ray. I'll see you next time on Catch This.